Hello. Advances in neuroscience have come from many areas of science and technology and engineering. Two of the most fruitful contribution areas have been drugs and toxins over hundreds and hundreds of years, and more recently, genetics. This lecture combines those two streams to talk about the genetics of nicotine addiction and recent data in that area. So we have finally arrived at the genome, a place where biologists go again and again. When the original nearly complete map of the human genome was first published in the year 2000, companion papers reported on the so-called HapMap project, which is a map of human genome sequence variations containing single nucleotide polymorphisms. These are places where one person's sequence may differ from another's, usually with no functional consequences at all. In the intervening years, the map has increased in density. It now has a mapped SNP every 1,000 bases, every 1 kilobase or so. So let's see how this resource helps to identify biomedically important genes for diagnosis and therapy. We're going to take a hypothetical example of non-nicotine dependent people and dependent people. At one point in the genome, one polymorphism, which we will call locus A, 20% of the people have sequence A1, 80% of the people have sequence A2, differing by one nucleotide. And this is true for non-dependent and for dependent people. And so we would say there is no linkage between this locus and nicotine dependence. But at another hypothetical locus in the genome, non-dependent people have 40% sequence B1, 60% sequence B2, but in dependent people, the distribution between B1 and B2 is different. So this means that sequence B1, that locus B may be near a gene that biases the distribution that helps to cause nicotine dependence. So with that idea in mind, people have done genome-wide association scans, GWAS they're called, to identify single nucleotide polymorphisms that affect nicotine addiction. The definition of nicotine addiction is, of course, the Fagerstrom test, a score of at least 4 versus 0. Now, in this particular set of observations, the researchers used people who had a score of 0 but still had smoked more than 100 cigarettes in their lifetime, found it easy to quit. And so these people were not cigarette naive. They knew all about cigarettes. The area that I've drawn a blue box around is not the highest in the particular study noted here, but this area has been replicated now in many studies, in many groups, for many aspects of nicotine addiction. And as you'll see, it's an extremely interesting part of the genome. So let's look in more detail at this area. Now we pass from a GWAS approach to a candidate gene approach, where researchers are pretty sure that this gene is interesting, and so they wish to study it more carefully. The candidate gene approach involves chromosome 15, about 3/4 of the way along chromosome 15, and we are going to expand this region. We find an extremely interesting set of sequences along this region. The region is about 100 kilobases long, so it's a small fraction of the chromosome. It has 3 genes and only 3 genes in it. Interestingly enough, it's a cluster of genes for nicotinic receptor subunits. The name of the gene, CHRNA5, cholinergic receptor nicotinic alpha polypeptide 5. So the gene encodes the protein called the alpha 5 subunit. The gene next door encodes the alpha 3 subunit. The one after that controls the beta 4 subunit. They are transcribed in both directions depending on the gene. So we have N to C for alpha 5, N to C for alpha 3, N to C for beta 4. Down here we have the log p-values for the probability that a SNP in this region is closely tied to nicotine addiction. The genes, as usual, have introns, which are these jogs here, and exons, which are much briefer bars. These are the actual regions that encode parts of the protein, and the introns get spliced out before translation. So, the region that is generally considered most interesting is in this exon. It's covered by the symbol for the polymorphism. A rather high probability, p-value less than 10 to the minus third, of being associated with nicotine dependence. So there are 2 possible base pairs at this locus, A or G. And if about 30 to 40% of chromosomes have the A, but a person has 2 chromosomes, and if both chromosomes have A on them, the person is twice as likely as other genotypes to score 4 or greater on the Fagerstrom test, abbreviated FTND here. So this is an extremely interesting region, whereas many of the other SNPs associated with smoking probably control the amount of gene expressed or are in regions that we don't understand. This is actually a polymorphism that's in a coding region and therefore presumably encodes a difference in the protein. Let's see where it is and what it tells us about the mechanism of nicotine addiction. Here is our usual diagram of a nicotinic receptor. We have the pentameric structure. We have alpha subunits, beta subunits. We have nicotine binding at the interface between the subunits. We have a cross-section here. Interestingly enough, the alpha 5 subunit, the one that is the genomic hit, is an auxiliary subunit. It is not at the binding site for nicotine. So that complicates things right there. There's nothing obvious about the alpha subunit. But let's look at the structure of the alpha subunit to find out where this interesting polymorphism is in which amino acid. Well, here it is. Oh dear. The interesting amino acid is number 398, and it is actually in a region of the receptor, the intracellular loop, where we do not have detailed atomic scale structure. And that's why I've grayed out this region. So we actually have to guess what is happening. We know that the amino acid encoded at position 398 is either aspartate or asparagine, depending on which allele a person has. We also know that the alpha 5 subunit can take its place in 2 types of nicotinic receptor. You'll remember that the alpha 5 subunit is in a cluster with alpha 3 and beta 4, and in many cases in the nervous system, the auxiliary subunit can be alpha 5 in an otherwise alpha 3 beta 4 receptor. In other places in the nervous system, the alpha 5 receptor can be in an auxiliary position in a receptor that has alpha 4 and beta 2 subunits. Usually we consider the alpha-4 beta-2 receptor as the one that is activated or desensitized at moderate levels of smoking. But we certainly cannot rule out the alpha-3 beta-4 receptor. So let's go a little bit further. The question becomes, what does this hit tell us about the mechanism of nicotine addiction? In particular, will it enable us to decide between the inside-out and the outside-in mechanism for nicotine addiction? So, a possible outside-in mechanism for this polymorphism, which we abbreviate alpha-5 D398N, is that although we don't have the structure, it seems to be in a region of the receptor that's near side windows in the receptor. And so when the current flows through the receptor, it actually flows through these side windows. And so one hypothesis is that position 398 actually governs the calcium permeability of the conducting pore. Well, that would certainly be important for the outside-in mechanism. And there are some data suggesting that the D398N polymorphism does control calcium permeability. Well, what about the inside-out mechanism? There's also a possible role for position 398 in the inside-out mechanism. Here is our diagram of nicotinic receptors leaving the endoplasmic reticulum, interacting with the COP2 vesicle. Here is a schema of the nicotinic receptor, these red bars here, interacting with the so-called Sec23, Sec24 complex. If we expand it, again, the structure is not known, but another hypothesis is that this region of the receptor is in approximately the right place to interact with the proteins that export nicotinic receptors from the endoplasmic reticulum, putting it in a crucial area for the inside-out mechanism. So this is about as far as we can go at the moment in our increasingly reductionist explanation of nicotine addiction. We have lots more facts to gather. Researchers are busy understanding alpha-5 D398N, and I'm sure that exciting data will be resulting. Next time, however, we'll talk about drugs for smoking cessation and harm reduction.